Let's talk sample prep. Of course, if you have a chemical reaction that's going to occur spontaneously, you can always mix the guest species together right inside the matrix box. Um, you could also do pyrolysis, which essentially is cooking them. You can do electron bombardment experiments, you can do photolysis, and you can do a bunch of other stuff. All kinds of neat stuff has been done with lasers and the like. Here is a large, although probably incomplete list of different detection methods that you can use with the matrix isolation technique. Although the staple is using an FTIR, you could also use a Raman, NMR, EPR, ESR, ENDOR. You've also got your UV-Vis, Mossbauer, as well as XAFs, which actually turns out to be a really important one if you want to find out any kind of bond length information. You also have some of your magnetics in there, MLD, MCD, MCPL. You could also do nuclear quadrupole resonance as well as INS, which stands for inelastic neuron scattering, which is a pretty obscure technique. This list definitely shows the diversity and versatility of the technique. As with any technique, there are challenges. Matrix isolation is no different. The first I briefly mentioned before is lack of bond length information definitely plagued a lot of early experimenters that use matrix isolation. And a lot of scientists these days tend to do simultaneous spectra in order to compensate for that. Also, in most cases, you're unable to actually trap excited state molecules in a matrix, mostly because they would have to actually be generated inside the box. And some scientists have figured out ways around this, and we'll take a look at some of that a little bit later. But also a lot of publications are met with scrutiny simply because there was originally a lot of guesswork associated with matrix isolation because they didn't have a whole lot to compare to. But nowadays, most scientists use ab initio calculations, which actually uses density functional theory to do huge integrations with programs like Gaussian. These are the kind of things where you tell the computer to do the calculation and you come back the next day and hopefully it didn't crash and it's all done for you. And so that will actually predict the different bands that you might see in a spectrum so that once the experiment is done, you have something to compare your findings to. Alright, we've reached the brief case studies section of the presentation, so stick with me, these can get a little complicated. First we'll take a look at a nickel ethylene system. It was originally investigated under matrix isolation conditions in 1992 with an FTIR, and the group found that there were 4 bands. But later in 1993, another team actually did the DFT calculations and predicted 12 bands for these same molecules. So another group went back and reinvestigated this in 1996, and by this time, higher resolution instruments were more readily available, and they actually were able to detect some of the weaker yet still characteristic bands that were actually predicted by the DFT calculations. And in addition to that, using deuterated species and so on, they were able to actually calculate the bond force constants for this. So this example really shows how important doing your own ab initio calculations can be whenever you're doing matrix isolation studies. So instead of this big long process where you may not know what to look for if you don't do these calculations, if you have all that data ready when you do your experiment, you're going to be in a lot better shape when you publish. Here's another case study where iron oxides were actually studied in an oxygen and argon matrix. They were annealed to a cesium iodide window at 10 Kelvin, and 3 distinct IR bands showed up at the values listed. And then they turned around and they annealed them at 50 degrees Kelvin on a similar window, and it actually turns out that there was a new IR band at 956 inverse centimeters. The new band is actually attributed to an oxygen-oxygen vibration, and the OO stretch is evidence of an eta-2 iron oxo system. And so the major conclusion is drawn that the molecule formed at 50 Kelvin is this complex iron oxo species, which was actually a really exciting formation. It was one of the first iron oxidation states of 6, which is pretty neat chemistry that's actually modeled after some of the heme and enzyme chemistry that you'll see. This particular case study really brings to light how much a scientist can really tailor an experiment to poke and prod these molecules until you get them to react just the way you want them to.